Okay. Assalamu alaikum everyone. My name is Malak. I'm a fourth year medical student, and today I'll be explaining to you guys gluconeogenesis and glycogen metabolism. Throughout the lecture, if you think I'm speaking too fast or if you want me to stop and repeat something, please do not hesitate. Also, the chat is open. I can see it. Okay, you can either unmute or use the chat. So first, we'll start with the gluconeogenesis lecture. Okay, so this image is actually very important, but we want to first understand it. So the thing is, we have three sources of glucose in the body or energy, tamam? First, we have exogenous, which is your diet, or you have your glycogen metabolism, or you have gluconeogenesis, tamam? Okay, so say for example, you eat a protein bar or a granola bar or whatever. Okay, so this will most likely last you 4 hours. Okay, it will peak and then it will last you 4 hours. After the 4 hours, if you're still fasting, what will happen? We'll start using like the glycogen. Now, after prolonged fasting, that's when gluconeogenesis will peak. So, you know what I mean? So first we start with the diet, then glycogen, then gluconeogenesis. Okay, so now let's just start with the basic information about glucose and glucose homeostasis. So we have the brain, RBCs, kidney, medulla, lens, cornea, testes, and exercising muscles. They all require continuous supply of glucose. Does anyone know why? Okay, so it's basically because they lack the specific enzymes for them to make their own glucose. Tamam? So these pathways are promoted by glucagon and inhibited by insulin. So as we mentioned here, it's because of the hunger. So will release glucagon and then we'll start making our own glucose. Also, this the liver maintains the glucose levels in the blood during fasting, either through glycogenolysis or gluconeogenesis. During an overnight fast, the gluconeogenesis occurs 90% in the liver and only 10% in the kidney. However, if, for example, you're stranded in the desert, if you're stranded in the desert, it's prolonged fasting, so then the kidneys will take over from the liver. Okay, now starting with actually gluconeogenesis. So first we'll start dividing it into three parts, the word itself. So gluco and then neo and genesis. So what do we mean by gluco? We mean glucose, and then neo we mean new. So genesis is the formation. So we're basically making or forming new glucose. Now, so as we mentioned, it's basically we're building something up and using energy. So can you guys tell me if this is an anabolic or a catabolic pathway? Okay. Exactly. It's an anabolic pathway. So how can we differentiate it? The way I think about it, for example, catabolic, think of a cat. The cat, they love to eat. So they break down the food and then when they eat, they start running around because they have so much energy. So catabolic, they break down stuff and they give us energy. And then anabolic is the opposite. I don't have a mnemonic for it. Okay, anyway, so it's a metabolic pathway results in the generation of glucose from certain non-carbohydrate carbon substrates. Keep in mind the carbon, and Hala will get to it in a second, but make sure that you know that it comes from carbon substrates. Okay, and as we mentioned, it occurs in the livers and the kidneys, but gluconeogenesis occurs in both mitochondria and the cytosol, except for glycerol. If the substrate was glycerol, معناها it will only take place in the cytosol. Okay. Now, another thing we want to talk about, which is the substrates. Tamam? So first, we're going to start with the glycerol, which comes from the lipids and the fatty acids. And as you mentioned, it only takes place in the cytosol. The lactate, which is, for example, from exercising muscles, we can transform lactate into glucose and bring it back to the muscles to use it. And we'll get to it in a second. This has its own cycle. For pyruvate, it's basically, sorry, one second. Yeah. So so the pyruvate, it's basically the reversal of glycolysis, which is the most basic, most common one. And then glucogenic amino acids, هلا we'll see the slides. Okay, one second. What's going on? Am I still sharing the screen? Yeah, okay. And then for glucogenic amino acids, so we mentioned إنه alanine is the major one. هلا it has its own cycle حتى, so we'll get to it كمان in a second. And we know our 20 main or major amino acids. What will happen here إنه all of it are glucogenic except for two, which are leucine and lysine. You guys can see هنا it's color coded. coded, sorry. So we have glucogenic, which is all the red, and then ketogenic, which is only these two, leucine and lysine. And then we have both glucogenic and ketogenic, which is the rest. So basically 18 of the 20 you can make glucose with them. Okay. Now, for other sources, basically we have dietary fructose and galactose can also be converted to glucose in the liver. In humans, it is not possible for us to make glucose from fatty acids. And why is that? Because هلا احنا we said إنه we can give بس we just said إنه glycerol can be used as a fatty acid. So sorry, can be converted into glucose. But why is fatty acid not? Because fatty acids and triglycerides or glycerol are not the same. Fatty acids are like a chain, okay? But the thing is, when you sorry, one second. So the net conversion of the carbons, as we mentioned, we need the carbon from fat into carbons. It's used in the liver as energy. For the liver, most we said, look here at, sorry, one second, look at the note over here. So we said, glucose produced by the hepatic glucogenesis does not represent an energy source for the liver. For what does the liver use instead of the glucose? It uses the fatty acid oxidation. So the acetyl-CoA has the carbons, and then these carbons will be excreted as waste, as CO2 in the TCA cycle. But we cannot use the carbon in the fatty acid. We'll use it in other sources of energy, but we cannot use it for gluconeogenesis. Now, this is basically what we said. The ATP, the gluconeogenesis requires ATP, which is provided by beta oxidation in the liver. So the liver takes the ATP and acetyl-CoA, and then it converts it into pyruvate, and then we can have our own glucose. Okay, is that clear so far? Okay, so now we have. Okay. So now we have the actual steps. So we have glycolysis and we have gluconeogenesis. Okay. So we want to reverse glycolysis in order for us to go from pyruvate all the way to glucose. But the thing is, most of the steps are reversible, which are the ones over here. But we have 3 steps that we cannot reverse. Okay. So the problem is, there's 3 steps that we need to bypass by specific or special ways. For the first step, we'll have pyruvate kinase, and then we have the rate limiting step, which is phosphofructokinase, and then we have hexokinase. Okay, so now the stars is the rate limiting step for both pathways. Okay, now as we mentioned, irreversible steps, hexokinase or glucokinase. Can anyone tell me what is the difference between glucokinase and hexokinase? Yeah, one is for the liver only. Exactly. So the glucokinase is actually only for the liver, and hexokinase is for the muscle. And then phosphofructokinase and pyruvate kinase. And then, as we mentioned, there's 2 steps there's 2 steps. These 2 steps can be bypassed very easily by hydrolysis, by losing a phosphate. There's only 1 step that requires more work, which is pyruvate kinase, the first step. We'll get to it. Okay. So first, two of the bypass reactions involve simple hydrolysis, which is the removal of the phosphate, adding water, and no ATP is made. Now we'll start with the first step. actually gluconeogenesis. تمام? Okay, so first, because we said the first step is the hardest one or the one that requires the most work, so we're gonna actually divide it into 2 steps, which is HOECAN, pyruvate kinase. Now we're gonna break down into 2 steps. So the first step will be, we wanna bring pyruvate into oxaloacetate. Tamam? This will happen in the mitochondria. So how do you remember the enzyme? What do you wanna transform? We wanna transform the pyruvate. So we'll add pyruvate carboxylase to give us oxaloacetate. Tamam? All of this happens in the mitochondria. And now, but for order for us to activate or speed up this process, what do we need? We need 3 things, which is A, B, C. So ATP, biotin, and CO2. Now, can someone tell me what biotin is? Sorry, guys, 1 second. My bad. Okay, so the thing is, the first step, as we mentioned, pyruvate to oxaloacetate using pyruvate carboxylase. So how do you remember it? We just add the name of the pyruvate to pyruvate carboxylase. So we have carboxylase, and we need 3 main things. to see, okay, to activate it or make it faster. So we need ABC, which is ATP, biotin, and CO2. And as you guys mentioned in the chat, so biotin is a vitamin, which is B7, and it's basically, you're very hard to be deficient in. You don't need to worry about being deficient in biotin unless you eat a very weirdly amount of raw eggs. Okay, that's the only way. And it's a cofactor for most of the carboxylase enzymes. So if you see carboxylase, it's a hint for you to know that the biotin is working in with the carboxylase. Okay, so another thing that we need to keep in mind is that acetyl-CoA is an obligate activator of pyruvate carboxylase. Pyruvate carboxylase cannot work without acetyl-CoA. So we need acetyl-CoA to activate the pyruvate carboxylase. Tamam? Okay. Now moving on to our second. Okay, let's go to the slide first. So basically we have, as we mentioned, acetyl-CoA is an obligate activator of pyruvate carboxylase. Now how does this happen? Okay, so the way this happens is pyruvate can be transformed into acetyl-CoA using PDH complex, okay, which is pyruvate dehydrogenase complex, okay? If the acetyl-CoA, and we have a lot of acetyl-CoA, we will have a negative feedback over here, so it will stop this complex and it will go and activate pyruvate carboxylate so we can make more oxaloacetate and shift it into glucose, okay? Does that make sense? Okay, and now this is we call it reciprocal control system. Okay, أنا صراحة I didn't think this was important, but it was in the review slides. So just make sure you know that this is called reciprocal control system. Okay. Now, for the second step, we want to make oxaloacetate into phosphoryl pyruvate or PAP. تمام? So this reaction will go out into the cytosol using the Malak shuttle. How do you remember the Malak shuttle? We'll say M for M. So to leave the mitochondria, we'll need another M, which is the Malak shuttle taking it out to the cytosol. Okay? So again, we're gonna make oxaloacetate into phosphoryl pyruvate using PepCK or phosphoryl pyruvate carboxylase. Again, the names are like a bit easy, because you just put the first name and then carboxylase, and here we have the first name and carboxykinase. Okay? Inshallah, it's clear. Clear? Okay. Now we're moving on to our third step, which is the rate limiting step. Tamam? So the rate limiting step, which is fructose 1,6 bisphosphate going to fructose 6-phosphate. So what do we do here? We said the last two steps are the easiest step, and you can do it by hydrolysis. What do you mean by hydrolysis? We want to remove a phosphate from the group, and then we can dephosphorylate it معناها. تمام? So here, instead of having two phosphates, one on 6, we just have it 1 on 6. تمام? So how do you remember this enzyme? It's basically the same name as the substrate. We just add ase in the end, so we know it's an enzyme, because the suffix ase, it means it's an enzyme. So how do you remember it? So it's fructose 1,6 bisphosphatase to give us fructose 6-phosphate. تمام? Okay. Now, as we mentioned, it's by hydrolysis, so it dephosphorylates using water and no ATP is made. Okay? Now, for this step, this slide, it can look a bit overwhelming, but inshallah, it's super easy. The main thing that you need to know is, we need, as we mentioned, this is a rate limiting step for both. So you want to know, is it going to glycolysis or is it going to gluconeogenesis? So we have this substrate called fructose 2,6 bisphosphate. Tamam? It's an intermediate substrate. If you have high levels of it, you will activate glycolysis. You will activate phosphofructokinase 1 and you'll convert it into this and then go on with glycolysis. But if you have low levels of it, it will do the opposite and it will activate the fructose 1,6 bisphosphatase. Tamam? Okay. So now, as we mentioned, let's look over here. So if you know that phosphofructokinase, which is this one over here, it's an insulin state. Why? Because we know in glycolysis we do it in the fat state, صح? But just remember, insulin state dephosphorylated, and it makes the fructose 2,6 bisphosphate, which is the intermediate that we need for glycolysis. On the other hand, if we do fructose bisphosphatase, when it's phosphorylated, it's in a glucagon state, which is for gluconeogenesis, and then it will break down the fructose 2,6 bisphosphate and continue to do gluconeogenesis. Is that clear? Does that make sense? Will I repeat? Okay, inshallah that makes sense. Okay. Now, another thing that we need to remember over here, guys, if you can see this table over here, is it clear? Inshallah it's clear. Okay, if you see the table over here, we have another thing to activate or deactivate fructose 1,6-bisphosphatase, which is the one for gluconeogenesis. So if you have high levels of ATP, it will actually activate. بس احنا هلا we're thinking انه gluconeogenesis is actually in the hunger state when you're actually hungry. So how does it make sense? انه if you have a lot of ATP, you're gonna have a lot of you're gonna make gluconeogenesis. It doesn't make sense. But the thing is, remember what we said in the beginning, that the liver needs fatty acid oxidation and energy from it in order for it to go on and make glucose using gluconeogenesis. But this ATP here is in the liver. معناها the liver needs good amount of ATP in order for it to make gluconeogenesis, okay? And also this is the fructose 2,6-bisphosphate that we just mentioned and the AMP, okay? Does that make sense? And this is like a repetition of what we said. So fructose 2,6-bisphosphate, if it's little, it will go shifted to the fructose 1,6-bisphosphatase and then fructose 6-phosphate and then into gluconeogenesis, okay? Now, for the last and final step, so again, it's an easy step. The last two steps are hydrolysis, which is this one over here. So it's hydrolysis. It's easy, inshallah. So we're going to make glucose 6-phosphate into glucose using hydrolysis. Again, it's the same name as a substrate, just add A's in the end so that we know that it's an enzyme, okay? So again, we're going to use this enzyme to remove the phosphate bond and then we can have free glucose. طب احنا why do we do this step? If we have glucose 6-phosphate, طب what's the point? عادي, خلاص, we can use it. Actually, if the glucose is phosphorylated, it's trapped inside the cell. But you cannot actually utilize it or use it. But that's why we need to remove the phosphate bond over here in order for us to have free and active glucose. Okay? Now, this enzyme is super important. Why is it important? Because this enzyme is not found in the muscles. معناها the muscles themselves, they cannot make their own glucose. Okay? And then this step, one other special thing about it is that it occurs in the endoplasmic reticulum by glucose 6-phosphate translocase. How do you remember this enzyme that it takes to this endoplasmic reticulum? So again, we use the same name as the substrate, but we add translocase because we're like relocating it to somewhere else, which is the endoplasmic reticulum. Okay, does that make sense? I need to repeat anything from any other steps? عشان I can go back and then we have a quiz هلا. Clear? Okay. Now let's draw this together. Let's basically just say what we mentioned again. Okay. So the first step is pyruvate. What is the next step? What will we have here? Okay, close, not pep, but we said we need to divide this step into two parts. So the main product from this step, we will have pep, but the first one, exactly. So oxaloacetate. Okay, what is the enzyme used over here from pyruvate to oxaloacetate? Remember how we tried to name it together? Pyruvate carboxylate, exactly. Very good. Now, we said we needed three things in order us to speed it up or activate it. What do we need? Exactly, ABC, which is? Exactly, ATP, biotin, and CO2. Perfect, mashallah. Now, the second step of this step, what is it? What did we need? Need? And exactly, acetyl-CoA, which is obligate activator of pyruvate carboxylase. Now, what do we have over here? This is the one you just mentioned, which was PEP. Okay, exactly. So this one is PEP, which is phosphoenol pyruvate. Now, what is the enzyme here? We mentioned the name. It's super easy. Exactly. So PepCK or phosphoenol pyruvate carboxykinase. Okay. Now we have a bunch of steps that we don't really care about. Okay. Until we reach fructose 1,6-bisphosphate. Tamam. Now, fructose 1,6-bisphosphate, what do you want to make it into? We want to remove a phosphate. So that, if that helps, يعني. Okay, so we make it into fructose 6-phosphate using, as we mentioned, we just put the name and add A's in the end. So fructose 1,6-bisphosphatase, and then we jump to glucose 6-phosphate. And then how do we convert it into free glucose and active glucose? What do we need to remove? We need to remove a phosphate. So what's the enzyme used? Okay, so it's glucose 6 phosphatase. تمام? Exactly, glucose 6 phosphatase. Perfect. MashaAllah, that was so good. Alhamdulillah. Does anyone have any questions on this? No? Okay. Now, another part of the regulation, and we need to know, as you mentioned, the hormones that are activating or inhibiting this gluconeogenesis. So this is basically glycolysis, tamam? So if you have glucose, glucagon, it will inhibit the formation of phosphoryl to pyruvate. Okay, it will inhibit the step, which is the pyruvate kinase, which was in the glycolysis step. Tamam? Another thing that it does at the same time, it promotes the transformation of oxaloacetate into phosphoryl so that we can shift the reaction from glycolysis into gluconeogenesis. Okay, and another thing that it does is that it inhibits the formation of fructose 2,6-bisphosphate, which what we mentioned. And if we have high levels of it, we will go into glycolysis. If we have low levels of it, we will have gluconeogenesis. Glucagon also inhibits the formation of this substrate, so that we can shift the reaction into going into glycolysis. Is that clear? Okay, now this is the easy part, inshallah. خلاص, we're almost actually done with the gluconeogenesis lecture. So the hormones, we have epinephrine, which raises blood glucose, activates glucogenesis and glycogen breakdown. Cortisol activates gluconeogenesis. Okay, and the way you can remember cortisol is that when you're so stressed, tamam, you have like a bunch of exams and you're studying. What do you need to do? And for me, personally, I take a break and I go eat something sweet. But think of it the same way. And the cortisol is a stress hormone. When the body is in so much, it's in stress, it activates gluconeogenesis to provide energy and glucose for the body. So another thing, and why do we have to keep this in mind? Because hyperglycemia is a common side effect of steroid drugs. So if someone was taking steroids, معناها they can have إنه steroid-induced diabetes. So we have to keep this in mind. Another thing is thyroid hormone, which activates gluconeogenesis. And that's basically it. Now we have the last two cycles. Okay, this is very easy, inshallah. هلا احنا we mentioned إنه the gluconeogenesis, the one we took, was from pyruvate. Now what if we have lactate? Okay, so think of it as the muscles. Okay, they're exercising. You have so much lactate building in the muscles, so much lactate. But as you mentioned, إنه the muscles, they lack the glucose 6 phosphatase. معناها they cannot make their own glucose. صح ولا لا? So what do? We have the lactate, the muscle sends it into the blood to the liver. بعديها in the liver will transform the lactate into pyruvate by lactate dehydrogenase. We have pyruvate, and then we have our own gluconeogenesis. We transform it into glucose, and then glucose to the blood, glucose to the muscle, and then we can use it by glycolysis. Okay? So it's really simple, and حتى it makes sense if you think about it. Lactate, liver, glucose, and then it goes back to the muscle as glucose to use it as for glycolysis. Now, this cycle is actually a bit expensive. Why? Because the liver spends 6 ATPs for a core cycle, and the muscles only gain 2 ATPs. فاحنا why are we doing this if it costs so much? Why can't just that we can use مثلا, for example, glycogen from the muscle? The thing is, this prevents lactic acidosis. Okay, فمعناها احنا we don't want to have a lot of lactate build up in the blood or in the muscles because it's bad for both of them. So what do? This Cori cycle basically prevents lactic acidosis. Now I want to ask you a question. If you have a defect in the Cori cycle, what do you think will happen? What are the two consequences based on this? Okay, so the two consequences, exactly. So it will be acidosis, exactly. So the first one will be lactic acidosis. The second one will be fasting hypoglycemia. لأنه حتى the muscles, they cannot make their own glucose, so they needed the glucose. Exactly. So fasting hypoglycemia and lactic acidosis will be a consequence of Cori cycle defect. Now, our last cycle for this lecture, يعني, we have alanine cycle. Okay, so the thing is, okay, let's think about it. So first in the muscle, we have amino acid breakdown. Okay, it forms pyruvate. تمام? But then as a consequence of all the reactions, we have NH3. NH3 can be toxic for the muscles and for the blood. So we don't want to release it into the blood to be excreted in the liver. Our body is actually so smart. So what does it do? It brings pyruvate and NH3 using alanine transferase, and it gives you alanine. Now, alanine is an amino acid that's not toxic عادي, so it can go into the blood and it goes to the liver. Now, in the liver, we'll break it down again from alanine to pyruvate and NH3, and then the pyruvate will be turned into glucose by gluconeogenesis, and the NH3 will be excreted as urea, which is a safe excretion. فإحنا we didn't want to bring it into the blood and make it toxic for the blood and the muscles. That's why we changed it into alanine. We moved it, we transported the alanine safely, and then we broke it down again and excreted this substance. Does that make sense? Okay. One second. So they combine pyruvate and yes, exactly. So احنا we have هنا we have the breakdown of the amino acid which gives us pyruvate and it gives us NH3. طيب فاحنا we don't want to have this toxic substance in the blood. So what do? We transform it into alanine, which is a safe thing that goes into the blood, and then we go into the liver. We retransform it back so we can excrete this safely in the liver. Does that make sense? Okay, perfect. Now, our last slide for gluconeogenesis, which is metformin. So metformin is a medication used in type 2 diabetes, and it inhibits gluconeogenesis. I don't know if you guys took the diabetes lecture yet. I don't think so. But basically, in diabetes type 2, we have insulin resistance. So the body is not using the insulin. فإحنا we have a lot of glucose أصلا to begin with. Since it cannot recognize insulin, they might want to go back and do so many other steps. فإحنا خلاص we go with metformin and we stop the gluconeogenesis. Okay? That's the only thing you need to know for metformin. Clear? Okay, so that was actually all of our gluconeogenesis lecture. Alhamdulillah, we finished it so fast. Any questions on the gluconeogenesis lecture? I can go back already. No questions? Perfect. تمام. Now, as we mentioned, we have three sources of glucose, which is exogenous, glycogen metabolism, and gluconeogenesis. So, as we mentioned, gluconeogenesis can take a long time. So, what do we start with? We start with glycogen. تمام? Okay. Now, these are the steps. They can look a bit overwhelming, but I can help you, inshallah, memorize it very well. Okay? Okay, so the first step, we have glucose to glucose 6-phosphate. تمام? Now, we know the step, which is glucokinase by annexokinase. It's the first step as glycolysis. تمام? Okay. Now, we want to transform, as you mentioned, in glucose 6-phosphate could be used into glycolysis. طب احنا how do we tell the liver? هلا I'll get to the questions, the practice questions in a bit, and I'll tell you which ones are the ones that come in the exam. Okay? But for the glycogen lecture, mostly glycogen synthase, branching enzyme, and then glycogen phosphorylase in the glycogenolysis and the diseases. Okay? Okay. Now for glucose 6-phosphate, we want to mark it because we said we can use a glucose 6-phosphate in the glycolysis. So how do we tell the liver, we don't want to use this for glycolysis, we want to use it for something else. What do? We actually rearrange the phosphate so it goes into glucose 1-phosphate instead of it being glucose 6-phosphate. Now, how do we mention the name of this enzyme? How do you use the name of this enzyme? Basically, we said we want to mark it, صح? And what do? We rearrange the phosphate on the glucose, تمام? Okay, so we have phosphoglucomutase. Now, this is not scientifically correct, تمام? But I'm gonna tell you the way I memorized it. And we want to put as if there's a mutation on the glucose 6-phosphate to change it into this, so that the liver knows that this is for glycogenesis, تمام? So we do the phosphate bond on the glucose to mark it as a mutation for the liver to know that this is going to glycogenesis. Okay? So this is how we memorize the enzyme. Does that make sense to you guys or just to me? Okay, inshallah it makes sense. Now another thing, هلا خلص we're starting into the actual glycogen. Okay, perfect. Now we're starting into actually the pathway of the glycogen. تمام? Okay. Now we want to change the glucose 1-phosphate since خلص we marked it and we're actually in the pathway for the glycogen. We're gonna change it into UDP glucose by UDP glucose pyrophosphorylase. inshallah it's the same, and basically you just add this name and pyrophosphorylase. Okay, now خلاص we're actually making glycogen and we want to elongate it. تمام? For what do? UDP glucose plus primer, which is glycogenin, as you guys can see, if you guys can see, yeah, here, glycogen, and this causes a linear chain by glycogen synthase. So glycogen synthase is actually the rate limiting step in glycogenesis. تمام? So we want to make a linear chain. هلا, can we stop here? No, we cannot stop here. Why? Because the body cannot store linear glycogen. احنا for us in the human body we need branches. The only linear glycogen is only in the plants. فاحنا خلاص we can't stop here. We need to continue. We create branches using branching enzyme. This enzyme inshallah is easy. Okay? So as we mentioned, glycogen synthase cannot start a glycogen change. It only extends one. Okay? If there is no glycogen fragment in we're supposed to add on it, we use the primer, which is glycogen. Okay? And glycogen synthase, even if it makes glycogen, it cannot give you the branches. It can only give you a linear chain. تمام? Okay. Now the branches in glycogen. Okay. So basically we wanna again, we said we're gonna market to make glucose 1-phosphate into the glycogenesis, صح? Now, since we're already in the pathway, we can't actually use glucose 1-phosphate as the base. We need UDP glucose. تمام؟ UDP glucose is what we need plus the primer, which will make us the glycogen. عرفتوا؟ the glucose 1-phosphate was only the start of the pathway. Does that make sense? Okay. Okay, now for the branching enzyme, what does the branching enzyme do? So the bonds are usually the linear chain by glycogen synthase is actually alpha 1 to 4 linkages. تمام? But the branching enzyme, as you mentioned هون, it makes amylose, which is in the plant tissue only. And the branches are by branching enzyme from amylo alpha 1 to 4 to alpha 1 to 6. So this is actually the branches. And you need to know the numbers 1 to 6. تمام? Is that clear? Okay, now I'm gonna quiz you. So does anyone have any questions on this pathway? No questions? Okay. Yalla, let's draw it together, guys. This is my favorite slide, by the way, the drawing. Okay, so we were gonna start with glucose, and we said the first step is the most basic one, the one that we can use for glycolysis. So what do we have over here? Glucose 6-phosphate, exactly. And what is the enzyme over here? Okay, so it is. Oh, exactly, hexokinase or glucokinase, depending on where do we do it. Okay. Now we said we wanna rearrange the phosphate home so we can mark it as something for glycogen. So what do we want to make it into? Glucose 1-phosphate, exactly. And then what is the enzyme used? Remember the three parts that we divided it into? Exactly, phosphomutase something, exactly. So it's basically phosphoglucomutase. So you want to arrange the phosphate group on the glucose to make it as a mutation, and we know this one will go into the glycogen. Clear? Okay, oh, okay. Now what is the next step over here? And I gave you the answer. غلط. So what is the answer? What do you want to change it into? Glucose 1-phosphate? UDP glucose, exactly. And which enzyme do we use? Exactly. So it's pyrophosphorylase, but very good, mashallah, that you know the name. Now, as we mentioned, and we need to add something here. What do we add to it so we can exactly glycogenin, exactly. So here we have the primer or the glycogenin, and now we're starting to elongate the chain using what was the linear chain, the elongation. What do we do? Yeah, it's 1,4, exactly. But what was the enzyme called? Okay, exactly. So it's glycogen synthase. هلا amylose, exactly. That's the name of the product, which is the alpha 1,4 bonds. This is what we call amylose. But the enzyme itself is called glycogen synthase, which is what makes the alpha 1 bonds, alpha 1,4. Does that make sense? Okay. Now, we have the simple chains. We wanna make it into more branches. What do we use? Exactly, branching enzyme. Perfect. And now the final product, what is the number of the bonds? Exactly, alpha 1 6. Perfect. Any questions? هلا we'll go into glycogenolysis. Unfortunately it's not as simple it is simple but it's not as direct as the reversal of glycolysis and gluconeogenesis. You know what I mean? This has a lot a bit more steps or different names of the enzymes. فهلا the thing is we want to degrade it into the primary product which is glucose 1 phosphate تمام. And also we want to break the linkages and the bonds. Okay, so now the first step, which is glycogen phosphorylase. Now this step is if I'm not mistaken, yeah, it's the rate limiting, exactly, rate limiting step of glycogenolysis. Okay, so we want to start احنا with the easiest part. We want to start with breaking down the alpha 1 to 4 linkages, which is the simple the simple linear chain. So we start with glycogen phosphorylase, تمام? And this releases glucose 1 phosphate. Now, as we mentioned, glucose 1 phosphate, it has a phosphate. So can we use this إنه as normal glucose ولا is it phosphorylated؟ معناها is it trapped in the cell ولا is it free glucose؟ Okay, exactly, it's trapped لأنه it has a phosphate group it's phosphorylated فit's trapped. تمام. So now as you mentioned, this is the rate limiting step. Glycogen phosphorylase catalyzes using phosphate, and the glycogen phosphorylase cannot break down the alpha 1 to 6 bonds. And this can only break down the simple chains which is alpha 1 to 4. You cannot break down the alpha 1 6. تمام. You cannot. This has another step, which brings us to step 2, which is debranching enzyme. فإحنا we started with the easier part, which is removing of the alpha 1 to 4, which is the simple linear chain. Now we want to remove the branches. Now this name is easy, debranching enzyme. Okay? فإحنا we want to remove the branches, which is debranching enzyme, alpha 1 6 glucosidase, and we remove it. We hydrolyzes the bonds, releasing the single residue branch. And this one, it gives us free glucose, much like this one. This one gave us glucose 1 phosphate, which was trapped. This one gives us free glucose, but you can use it على طول. Okay? And this represents the only free glucose produced directly in glycogenolysis. Clear so far? Okay. Now, our final step in glycogenolysis, it's basically phosphoglucomutase. If you remember the step, this is the same exact step that we used to make glucose 6 phosphate into glucose 1 phosphate. صح ولا لا؟ what we said we mark it to make it mutation. Exactly. So glucose 6 phosphate to glucose 1 phosphate. Now it's the same enzyme so that we can reverse it back. But since إنه خلاص this is the breakdown for the enzyme is tired, but we break it down into 2 steps so it's easier for the enzyme to work with. So the first step will be glucose 1 phosphate to glucose 1 6 bisphosphate. So this is like an intermediate substrate. And then we bring the glucose 1 6 bisphosphate to glucose 6 phosphate. Clear? Okay, now since our end product is glucose 6 phosphate, as we mentioned in the last step of gluconeogenesis, and what happens in the endoplasmic reticulum, so again, we'll use the same enzyme we used over there, and we'll take it into glucose 6 phosphate translocase because we're relocating the enzyme into the endoplasmic reticulum, and we'll start to convert it to glucose by glucose 6 phosphatase. Does that make sense? Okay, inshallah no questions. And هلا actually we're done with glycogenolysis. هلا we're just going to talk about the regulation. خلاص we're almost done guys. تمام. Now for allosteric regulation أنا صراحة I even had a hard time like understanding what allosteric even means. No, I didn't get it. But the way I think about it is, so for example, think إنه we have you have your TV at home and you have the screen which has buttons in the back and you also have the remote. ف if you're doing it directly from the screen, this is not allosteric. But if you're using the remote to control it and to turn it on, turn it off, speed it up, or whatever, this is what we call allosteric regulation. So it's basically of binding of something, but not in the same place where it's supposed to bind. Like it activates it somehow, like it does like so many other reactions and activates it or inhibits it somehow. So glycogen synthase and glycogen phosphorylase respond to level of metabolites. As we mentioned, if you have a lot of this يعني for example, glycogen synthase stimulated by glucose 6 phosphate. If you have a lot of glucose 6 phosphate, you will shunt it and make it into glycogen. On the other hand, if you have high شو اسمه sorry low glucose 6 phosphate and other يعني it will إنه inhibit the glycogen synthase. So this is basically يعني what it's talking about. Now for glycogen degradation in the muscle, so say the muscle is contracting تمام and there is rapid need of ATP. We need the ATP. خلاص there's not enough oxygen, there's not enough energy. خلاص the muscles are. So now this sentence over here, this is basic physiology, which is the membrane potential and all this stuff. So the nerve impulses cause membrane depolarization, which promotes calcium release into the sarcoplasmic reticulum, into the sarcoplasm. تمام? So we started with the basic physiology. After that, the calcium will bind into calmodulin. Then the calmodulin, when it's bound to the calcium, it will make its own complex, which is calcium calmodulin complex. Then this complex will activate phosphorylase kinase, which will then activate glycogen phosphorylase, which will activate the degradation. You know what I mean? So it's not as direct as we think. There's like so many steps for us to go around, and then we activate the glycogen phosphorylase. Do you get what I mean? In this slide, will I need to repeat anything? Okay, I can repeat, of course. Okay, so say we have muscle contraction, tamam? The muscles are contracting there. We have action potential, depolarization, and the muscles right now, they need the ATP. They need glycogen. They need glycogenolysis to utilize this, tamam? So the nerve impulses, what do we have? We have depolarization. So it will promote the release of calcium release from the sarcoplasmic reticulum into the sarcoplasm. Tamam? Is that clear so far? This is basic physiology. Yeah. Okay. So now after that, the calcium will bind to something called calcium binding protein, which is calmodulin. When the calcium binds to the calmodulin, we'll have a complex called calcium calmodulin complex. Okay. Now this complex will directly activate phosphorylase kinase, which is not the thing we know, which is not glycogen phosphorylase. It's something else. So this phosphorylase kinase will go and activate the glycogen phosphorylase so we can have glycogenolysis. Does that make sense? Will I repeat again? عادي, I don't mind. Good? Okay. Now again, this is another note إنه muscle glycogen phosphorylase is active in the presence of high AMP. إحنا we need ATP. But when you have high AMP, إنه لا خلاص we have ATP depletion and anoxia. We need to go and start the degradation. تمام? Okay. Now we have to pay attention and there's a lot of steps إنه إن شاء الله it's simple but إنه it's a lot of steps so we have to pay attention to it تمام؟ Okay. So the first step, which is activation of protein kinase. So this is a protein kinase, it's inactive and it has it's a tetramer, okay? So it has four things. So it has two regulatory and two catalytic, تمام? And the activation is dependent on CAMP. So when CAMP, which is cyclic AMP, it comes and it binds to the protein protein kinase, it binds to the regulatory, it binds the regulatory and it releases the catalytic. معناها this is when it becomes active. But when we remove the CAMP, it becomes inactive again. فit needs the CAMP to be activated. Is that clear so far? Then we're building like a pathway. It has three steps. Okay. It has three steps. So this is the first step. We activate the protein kinase by binding of CAMP and we remove and then we release the شو اسمه releasing the individual active C subunits. Okay? Now, the second step, which is activation of the phosphorylase kinase. Okay? Now, we said the protein kinase, this is active. So this one goes and activates phosphorylase kinase. تمام? Okay. The thing is here, I want you guys to pay attention and phosphorylated means active, dephosphorylated means inactive. But the thing is, there's another enzyme that we'll get to it in a second where it's at the opposite. But how do you memorize it? If you see phosphorylase, this is not a spoken rule, but in this here now, this is it's related. If you see phosphorylase, the word phosphorylase or phospho, معناها when it's phosphorylated, it will be active. So phosphorylase, phosphorylated, phosphorylase, phosphorylated. Okay? Is that a good mnemonic or am I confusing you guys? Okay. Okay. So again, if you see phosphorylase, معناها when it's phosphorylated, it will be active or A form. Dephosphorylated will be inactive. So what happens? The active protein kinase from the previous step, it goes to the phosphorylase kinase. It phosphorylates it to make it active. Okay, this is basically what the step is talking about. تمام. So we took this step, we activated it, and then we go using the active protein kinase, we go and activate phosphorylase kinase by phosphorylating it so it's active, so it's A form. And then this is the second step. تمام. Now for the third step, we have glycogen phosphorylase. Again, the same name, phosphorylase, phosphorylated, so it's active, so it's A form. تمام. Dephosphorylated is inactive in B form. And this does the same thing. So let's go back and recap. تمام. First step, we have CAMP binds the protein kinase, it activates it. The active protein kinase will go into the phosphorylase kinase, phosphorylated, make it active, and then we have the phosphorylase kinase will go and activate the glycogen phosphorylase by phosphorylating it. One second. So after CAMP binds activated, basically, exactly. Wait, let's go back. Exactly. So it goes and phosphorylates the inactive form of phosphorylase kinase, leading to activation. فاحنا we have three steps basically. Two of them are exactly phosphorylating. So again, let's recap one more time. Protein kinase, CAMP, activating it. It goes to phosphorylase kinase. It activates it by phosphorylating since it's in the name. After that, the phosphorylase kinase goes to glycogen phosphorylase, phosphorylates it again, so it's active. Clear. Okay, alhamdulillah. تمام. Now هلا هون this one was for the activation of the degradation. But what if احنا we just want to inhibit the glycogen synthesis by the CAMP? Okay, so this is regulated by glycogen synthase مش glycogen phosphorylase. This one was, as you mentioned, it was a step in the degradation. This is a step in the synthesis. فاحنا we want to stop the step. What do we do? هلا as I mentioned to you guys, that's why I told you to pay attention. لأنه phosphorylate here, if it's dephosphorylated معناها it's active. So you know how it's the opposite? Because it doesn't have phosphorylase in the name. So when it's dephosphorylated, it will be active. Does that make sense, برضو? Okay, exactly. So then the glycogen synthase is converted to the B form, which is by phosphorylation. So it's inactive. And then we can, the conversion process is by protein kinase regulated CAM. Okay? So as you mentioned, in contrast to glycogen phosphorylase, glycogen synthase is inactive when phosphorylated. Make sense? Okay, now the second part of this step, which is the inhibition of the synthesis, we have this. I just wanted you guys to first, before we continue, and you have an idea that the glycogen synthase is activated by insulin in the liver and in the skeletal muscle. But it's inhibited by glucagon or epinephrine in the liver and skeletal muscle in the epinephrine. In the epinephrine in the skeletal muscle. تمام? Okay, so now this is basically a recap of what we said. تمام? We have a binding of glucagon or epinephrine in the hepatocyte, as you mentioned over here, or we have, where am I? Yeah, or we have epinephrine in the muscle cell receptor, activation of adenine cyclase, and then we activate the CAMP-mediated protein kinase, phosphorylation, and then inactivation. Does that make sense? Okay. هلا we're done with the basic biochemistry part. Alhamdulillah. Any questions on gluconeogenesis or glycogenesis or glycogenolysis before we start into the diseases? No questions? Tamam, we'll start. So now for glycogen storage disease, it's mostly genetic enzyme deficiencies associated with accumulation of glycogen within the cells. Okay, so today we'll be talking about 1, 2, 4, and 5. This was never mentioned in the slides. And this was never mentioned in the slides. Okay, so we'll start with von Gerke disease or glycogen storage disease 1. I recommend in you guys know the numbers. I remember in third year we had the questions like about the numbers, I think, but I'm not sure about first year. But it doesn't hurt for you to know the numbers, okay? Okay, so you have von Gerke disease. The problem with von Gerke disease is that we have a deficiency in the enzyme glucose 6 phosphatase. Because can anyone tell me? Oh wait, let's cover this. Oh, I can't cover. Anyway, can anyone tell me what is the step? Where did we learn it today? Okay, so basically we learned it in the gluconeogenesis lecture, which was the last step, which is from glucose 6 phosphate into free glucose. تمام. So if we don't have this enzyme معناها we're gonna have a lot of glucose 6 phosphate. Glucose 6 phosphate, as we mentioned, it's an activator of glycogen synthase. معناها it will shift the reaction. Instead of having accumulation of this, it will shift the reaction into making more glycogen. تمام. So what we'll have prolonged fasting hypoglycemia since we can't make glucose 6 phosphate into free glucose because we're lacking this enzyme in the liver. Also lactic acidosis and hepatomegaly. Now additional symptoms like glycogen deposits in the liver. So a glucose 6 phosphate stimulates glycogen synthesis and glycogenesis is inhibited. So we're gonna have a lot of accumulation in the liver. Also hyperuricemia. Did you guys take gout ولا لا? Okay, so basically gout is basically we have this thing in the body called we have this thing called uric acid. If we have a lot of uric acid, it builds up in the joint and it causes like the joint to be like super big and red, super big and red. تمام? Sorry, super big and red. It will cause hyperuricemia, predisposing to gout, and also we will have hyperlipidemia with skin xanthomas, elevation of triglycerides. Now, another problem we have is that ingestion of galactose and fructose causes no increase in the blood glucose. And the epinephrine and glucagon administration does not help. So it does because احنا خلاص there's no reaction for us to shift into because we're missing this step. You know what I mean? Is this clear? Okay. Now, I need you guys first to memorize this picture, and that's very important. Now we'll talk about the clinical manifestations. So as we mentioned, because we cannot make our own glucose, so we'll have hypoglycemia with lactic acidosis when fasting. Also, distended abdomen due to hepatomegaly because we have a lot of accumulation in the liver. Dull-like features or face. Poor growth, seizures secondary to hypoglycemia, again, because we cannot make our own glucose, they will have severe hypoglycemia. Also, increase in triglyceride and cholesterol. Okay, now how do we treat it? We give them continuous infusion of glucose, especially at night until the age of 2. Okay, and then until when they reach the age of 2, we can give them cornstarch. What is cornstarch? Basically, it's like something you eat. The body cannot digest before they're 2 years old. That's why we give them when they're 2 years old. We give them, and it's basically like a slow release of glucose. So it's basically like its own like regulation thing. That way, the body does not have very low glucose because if you eat cornstarch, it will start releasing it as glucose in a very long amount of time. So we'll have regulated blood glucose for a long amount of time. Does that make sense? Okay, and also we have frequent meals and snacks, so again we can prevent the hypoglycemia. And allopurinol is a medication for the gout. So if the uric acid is too high, allopurinol is the medication. It was in the slides. I don't think you should know it, but it's in the slides. And also we have a liver transplant later, in the later progression of the disease. Now I have this mnemonic for you guys, which is Van is stuck in the garage. Okay, so Van is for Von Gerke, and why is it stuck in the garage? So G is for glucose 6 phosphate, and it's stuck in the liver, accumulating in the liver. Okay, so Van is stuck in the garage. So Von Gerke has a lot of glucose 6 phosphate stuck in the liver. Does that make sense? Okay, any question on Von Gerke before I move on to the second one? Okay. Okay. So now we'll talk about Pompe disease. تمام? We have lysis. Sorry. Okay. So the deficiency is basically in this one is in glucose 6 phosphatase. فإحنا we cannot move glucose 6 phosphate into free glucose. When we have a lot of accumulation of glucose 6 phosphate, what will the body do? And we will shift the reaction into glycogen because the body thinks إنه إحنا we don't need the glucose. So إحنا we have a lot of this, so we'll shift it into glycogen production. For the deficient enzyme is glucose 6 phosphatase. Yes, exactly. We cannot move it into or transform it into free glucose. So we have a lot of glucose 6 phosphate. We transform it into glycogen and we store it in the liver. تمام؟ Okay, of course. Okay, as we mentioned. Now this is the mnemonic. Now moving on to our second disease, which is Pompe disease or lysosomal alpha-1,4 glucosidase deficiency or glycogen storage disease 2. Okay, now well, the problem is the lysosome itself cannot um, يعني break down the glycogen تمام. But if you look in the cytosol or the cytoplasm, the glycogen levels will be normal. But in the lysosome, they will be super high because they're lacking this enzyme, which is lysosomal alpha-1 glucosidase deficiency. Okay, so you're lacking this enzyme which will break the glycogen or the bonds in the lysosome. تمام. So we'll have, because look, I don't know if you can see this picture over here. But anyway, basically انه it's this is the lysosome انه it's you can see انه it's enlarged. تمام. Now, what is the consequence of all of this? We will have profound, rapidly progressive muscle weakness. So the baby will be like, if you carry him, it will look like a doll. There's no muscle strength. There's no tone. Okay. Also, cardiac, cardiomegaly, and cardiomyopathy. I'll come back to it. Respiratory progression to pulmonary insufficiency, hepatic hepatomegaly. Now, why is this in red? لأنه if left untreated in the first 2 years of life, the child will die because of cardiomyopathy. Okay, so this is very important and it's a keyword in your MCQs. تمام? All clear? Okay, now here again our mnemonic, Pompe or lyso pump disease. So lyso for lysosomal accumulation and pump means heart, so heart failure. And if you guys can see, look at the picture here, the baby doesn't have any tone. Like his muscles are so relaxed, he has so much weakness. Any questions before I move on to the third one? Okay. Perfect. Now, for Anderson disease, this is glycogen storage disease number 4. As you mentioned, we skipped number 3 because it wasn't in the slide. So number 4, also known as amylopectinosis. Why? Because we have a deficiency in the branching enzyme. فإحنا what do we have? We only have the 1 to 4, the 1,4 alpha 1,4 bond, صح? Which is the linear chain, as we mentioned. Which, as we said, this is the starch found in the plant, صح? Exactly. So it's also called amylopectin. So amylopectinosis معناها we have a lot of amylopectin accumulating in the body. تمام. The manifestation with its progressive hepatosplenomegaly alongside with abnormal glycogen. Okay. Any questions on this one? It didn't really have a lot of information in the slides. So this is what we basically have to know. Okay, and we have the mnemonic for it, which is A is for Anderson. So Anderson abnormal branches. Good. Okay. Now, our last disease for today, and خلاص, actually we're almost done, we just have the questions now. Okay, so McArdle disease or glycogen storage disease 5, also called as myophosphorylase. So basically, the presentation of this, you have an adult or say someone who's 20, tamam, they're walking outside, they're running, they're exercising, they have severe muscle pain after they're exercising, and then when they use the bathroom, they have burgundy colored urine. It's brown. Okay? So this is basically the manifestation or the scenario that will come to you in the question. Tamam? So about half of the the glycogen storage disease number 5, patients will exhibit burgundy colored urine after exercise. Now, for the mnemonic for it, McArdle, which is M&M. So for muscle cramps and myglobinuria. And you guys can remember this image because he's doing it too. So M&M. So myglobinuria and muscle cramps. Any questions? Okay, so now we're moving on to the questions. So if you want me to go back and explain anything, just let me know عشان we can move on to the questions. Is everything clear? Okay, inshallah, it's clear. So I'll read the question and then we can answer it together. Okay. So we have when it okay. We have what's going on? Okay. We have examining 5-month-old infant who was brought to your office by distressed parents. Okay, mashallah. Okay, their infant has demonstrated progressive weakness and difficulty eating and appears to have a bulging abdomen. Upon examination, you find striker hypotonia, palpable liver, chest rate indicates cardiomegaly. Before a correct diagnosis was made, the infant lapsed into a coma and dies. Which of the following is most likely the disease? So someone said D, the rest. Okay. Okay, can you guys tell me why you chose D? Sorry, E. Can someone tell me why you guys chose E so I can go back and we can explain it again? Okay, so I get what you're saying, but also we cannot only hang on to one clue and then not go to the rest. But another specific thing was, they're 5 months old and they had cardiomegaly, which is very specific for Pompe disease, and they went into a coma and they died. You know what I mean? So this is a very highly indicative of Pompe disease. Okay, and also we mentioned, one second, where's Pompe? Yeah, we mentioned and we have hepatomegaly, hepatic hepatomegaly. فالso we will have bulging abdomen in the Pompe disease. تمام, does that make sense برضه؟ In Bang, yeah. I think hepatomegaly بس. I don't think we have hepatosplenomegaly. Hepatosplenomegaly was in Anderson. This one. Hepatosplenomegaly. Okay? Inshallah, with practice, you'll memorize it more. Okay? And try to remember the mnemonics also. حتى the mnemonic was pump. It's very indicative that the heart cardiomegaly, you know? Okay. Now we'll move on to the question. So it's Pompe disease, okay? Any questions on this? Do I need to explain anything else? Okay. Now moving on to this question. So an increased levels of glucose 6-phosphate is expected to have which of the following effects on carbohydrate metabolism hepatocytes in hepatocytes? Guys, pay attention to this one. Read it carefully, okay? Okay, let me explain this one, okay? It is a bit tricky صراحة and I got it wrong in the first time. But okay, we'll say. So glucose 6-phosphate معناها احنا we have a lot of glucose 6-phosphate. So as we mentioned before, we'll shift it into glycogenesis تمام? So you want to make more glycogen. So saying the sentence, we can eliminate more than one thing. لحظة, let me bring the pen. Okay, so we can eliminate this. Why? Because in glycogen phosphorylase, this is for degradation. Also, glycogen synthase, we can keep this, we can keep this, we can remove the glycogen phosphorylase. Inhibition, okay, this خلاص, we'll leave it for later. Now, for me, I will go into these two. لا, why? لأنه احنا خلاص we want to make more glycogen, صح؟ فاحنا we need glycogen synthase. فwhat do we do? Do we activate the active one, which is A, ولا do we activate the inactive one, which was B? هلا, okay. Okay, هلا, wait, let me erase this and we'll go back again. Just erasing. What the hell? I actually do not know how to erase. Never mind, found it. What the? Okay, guys, I'll explain it on the answer slide عشان I messed up. Okay, خلاص, let me remove this. Okay. Okay, so let's think about it again. تمام? We have glucose 6-phosphate. احنا we mentioned if we have a lot of glucose 6-phosphate, we will shunt the reaction into glycogen metabolism, صح? We want to make glycogen, صح? Is this quite clear ولا لا؟ Okay, تمام. Now, glycogen phosphorylase, did we use it in the making of the glycogen ولا the breakdown of glycogen? Where did we use it? Exactly. So why would be activated now? احنا we don't wanna cause degradation. احنا because we have a lot of glucose 6-phosphate, we wanna build it into glycogen. So why would we activate the one that's breaking glycogen down? We don't want that. That's why I crossed it off. Does that make sense? Okay. Okay. Now هلا why did I put the options حتى this one برضو نشخطها. Okay, so why is it B and C? Because احنا we want to activate glycogen synthase so we can actually start shifting it into glycogen metabolism, sorry, synthesis. تمام؟ And glycogen synthase is the rate limiting step as we mentioned glycogenolysis. That's why احنا we needed to shift it into this reaction. Now, we mentioned in glycogen synthase A. So because we have too much glucose, we want to make it into. Okay. صراحة I don't understand what you mean inactive. هو it's already inactive, it's not free. يعني it already has a phosphate group. تمام. فإحنا we want to make it into glycogen, which is the glycogen synthase step. Okay, which is the rate limiting step. فإحنا we want to activate the step. هلا إحنا we mentioned that A is already active. It's already phosphorylated and active. فإحنا why do you want to activate the active? We will go and activate the inactive. Do you know what I mean? Okay. Okay. I still don't understand your question. No, listen. This glucose is not an active glucose. This is a glucose 6 phosphate. It's phosphorylated, so it's already trapped in the cell. فإحنا we can't use this except for gluconeogenesis or glycolysis or for glycogen. But since we have a lot of this, we don't want a lot of this. This is like an activator. If you have high levels of G6P, معناها it's an activator of the glycogen pathway. تمام؟ فإحنا what do? We want to activate the glycogen synthesis pathway. Do you get this part ولا لا؟ Okay, exactly. فإحنا we want to start activating the glycogen synthase because it's the rate limiting step of the glycogen synthesis. Okay? ف we said in glycogen synthase A is active, B is inactive. تمام؟ So, do you want to activate the inactive ولا do you want to activate the active؟ إحنا we want to activate the inactive so the inactive starts working. صح ولا لا؟ Exactly. So that's why you'll start activation of a glycogen synthase B. Does this question make sense? Or let I go back to the slide for it? تمام. Everyone else, does it make sense? Okay, inshallah, it makes sense. Perfect. Now, another thing, muscle membrane will depolarize. Guys, pay attention. It's a bit tricky. طيب. Muscle membrane will depolarize in response to acetylcholine binding its receptor at the neuromuscular junction associated with depolarization, changes in glycogen metabolism in skeletal muscle cells. Which of the following represents the correct changes in enzyme activity seen in response to acetylcholine binding? So think of it as the thing we mentioned, and we have a lot of calcium. I actually forgot the answer for this one. The rest, D. Let's find out together, guys. Exactly, D, correct. MashaAllah. Does anyone want me to explain this one? Okay, so we mentioned if you have a lot of calcium, it will bind to the calmodulin and it will cause a complex, and then this will start activating the phosphorylase kinase, which will then activate glycogen phosphorylase, which is the breakdown of glycogen. So if you see calcium, معناها it's an indicator that the muscles are working. There's a lot of depolarization happening. The muscles are contracting. But we want to make energy for the muscles. So we go to glycogen breakdown. تمام? So increased glycogen phosphorylase kinase activity. Good question. I don't know because this is basically physiology صراحة. I don't remember it. Sorry. Okay. So increased glycogen phosphorylase activity due to an increased calcium binding. Okay, let's go back to this slide actually. Mm, well, which step was it? This one? Oh, allosteric صح, my bad. خلاص, we're here. Okay, exactly. So I'll explain the slide again, okay? If you want me to, and then after that, if you don't understand, just let me know. Okay, so the question mentioned, there's so much there's muscle contraction, so we have depolarization and we have calcium release. The calcium then will go and bind to the calmodulin. When the calcium binds to the calmodulin, it will form this complex, which is calmodulin calcium complex. Tamam? Okay, now this complex will go and activate phosphorylase kinase, which will lead to high levels of phosphorylase kinase. This is what the answer said. High levels of phosphorylase kinase activate glycogen phosphorylase, leading to glycogen degradation. Does that make sense? Okay. Now let's go back to the slide. Start. Let's see. Okay, so Z. This is basically what we said: increased glycogen phosphorylase kinase activity, and due to an increased calcium binding to calmodulin subunit. Okay. Now this question. You are studying a cell line of gluconeogenesis and affected changes in the acetyl-CoA. Pay attention to acetyl-CoA. These cells most likely harbor an enzyme that, I can't read this word, allosteric, I can't see it actually, anyway. Lost its allosteric binding site for acetyl-CoA. What will happen? D, perfect. Exactly. So as we mentioned in acetyl-CoA, it's an obligate activator of pyruvate carboxylase. So we need it in order for this enzyme to work. Any questions on this one? Okay. I think this is our last question, if I'm not mistaken. Yeah. So during fasting, glycogen is used as a source. Guys, pay attention to this one. Pay attention. Okay. During early fasting, glycogen is used as source of glucose for the blood. After liver glycogen is depleted, which of the following occurs? Someone said A. What else? Okay. So why did you say C? Okay. هلا the thing is احنا we said in fatty acids actually we cannot convert them into glucose. Even the same thing here. Fatty acids are degraded as precursor. They're not used as a precursor. Okay? But exactly beta oxidation, correct. You're thinking in the right path, but not completely. So احنا we mentioned that the beta oxidation is the use of it will give energy for the liver in order for the liver to make the glucose. تمام? So it's not used as a precursor for the glucose. Do you know what I mean? So we cannot convert fatty acids into glucose in the liver. We only use the fatty acid oxidation or the beta oxidation for ATP for the liver to carry on the reaction. You know what I mean? Clear? Okay, so the answer is actually A. So the amino acid of the muscle protein are synthesized as glucose in the liver. Because we mentioned in the first slide, which was we had the substrate used, we had glycerol, we had the pyruvate, we had lactate, we had amino acids, and we mentioned that even glycerol is not the same as fatty acids because fatty acid can never, ever, ever be used as precursor for blood glucose. Okay, does that make sense? In general, yeah, that comes from the muscles or in general. لأنه it's not only always alanine. If you remember the first slide, مصح, we had 18 of the 20 18 of the 20 amino acids can be used as a precursor for the gluconeogenesis. فit's not necessarily يعني from the muscles. All clear? Perfect. Does anyone have any questions? لأنه الحمد لله we're done. Oh, where's the QR code? Okay, you guys can scan the QR code so we can do your feedback if you have any questions and so on. And here's also my number. You guys can contact me at any time. I can answer your questions at any time, inshallah. I'm more than happy to help. Does anyone have any final questions? Perfect. Thank you so much.